At this point, you've tried your class exercise on allele segregation. You should be able to follow alleles segregating during meiosis, and you should be able to do this for 2 chromosomes and 2 alleles. Let's move on now to the next topic, which you will see is highly related to allele segregation, and that is the topic of genetics and crosses. We've talked a lot about genes and gene variants that are caused by changes in DNA sequence. And that indeed is how inheritance works and changes. It's all about what the gene looks like at the DNA sequence level. But sometimes we really don't understand what the DNA sequences are that are giving rise to a particular trait. And then we need a different set of tools to help us understand something about how traits are inherited and how a particular trait might be regulated. And this is important across the board. It's very important when it comes to things like diseases, where we try to understand what it is that is causing the disease, even if we don't know what the gene or genes are, and even if we really think that there might be something very complicated going on. So, genetics is the set of tools to help us understand inheritance and gene function. Genetics, tools to understand genes and inheritance. These tools are what we need to discuss so that you can use the power of genetics to understand something about a particular question. And we need some definitions before we can move on. Let's make a short listing of some key points you need to know the terms for. Genotype refers to the genes of an individual. Phenotype, we've mentioned, is an observable trait that you can see by eye or by some kind of other assay. Gene, in molecular biology, we talked about the DNA sequence required to make the final product. In genetics, the definition is slightly different. It's a little vaguer. It's referred to as the hereditary unit. Allele, for completeness' sake, we'll write it again, are alternate gene forms. And then the last one we need is generation. And this refers to individuals that are born at the same time from the same parents. We can talk about the P generation, which are the parents. We can talk about the F1 generation, which are the first generation offspring. We can talk about the F2, which are the second generation offspring, etc. Okay, I'm abbreviating generation there with gen. Those are some of the key terms that we need as we go and we consider some of these observable characteristics. Okay? So let's think about some of the observable characteristics and some of the genetics that we are going to talk about by looking at this slide. Genetic traits can have the following terminology. They can be dominant, they can be recessive, codominant, or incompletely dominant. And we'll talk about some of those in a moment. But for example, you may have freckles or not freckles. Freckles is considered dominant. And if someone were to mate with someone who didn't have freckles, probably the offspring would be freckled. You might know about rolling your tongue. Some people can roll their tongue. I can roll my tongue, can you? Rolling your tongue is considered dominant. It's something that if two people get together and have offspring, then the children, or some of them, will be able to roll their tongues. In flowers, moving away from humans, there are flowers that are mixed colors. There's a dahlia that's red and white. That's an example of a trait that is called incompletely dominant or codominant sometimes, okay? And that, again, will give you a sense of how genetic traits can move around in different organisms. And this bottom example is very interesting. This is a sickle cell, a red blood cell that, with a particular gene variant, a particular allele, can change from a round cell or a disc-shaped cell to a kind of a plate-shaped, indented cell. And this has to do with a characteristic called recessive. And it's associated with various disorders, but can also be very useful in certain circumstances. So this gives you some sense of where we're going to go in this part of the lecture. And you may not have ever heard of these terms, and that's fine, because you will now. I've also given you a listing of some of these genetic terms that you can refer to in addition to what is on the board. Let us now consider genetic crosses. And we're going to start off with genetic crosses that follow a genotype. And particularly, we are going to talk about monohybrid crosses, which means that there's one single trait involved and likely one gene. Monohybrid cross, that means one trait, probably one gene. And we can write down this genetic cross in the following way. We'll write the parents, we'll write the genotype, we'll write what gametes they are going to form, and then we'll write what the first generation offspring are going to look like. So we're going to do a theoretical breeding experiment here. The parents are designated P1 and P2, and let us say that the genotype of the first parent is big B, big B. This means that that parent has the alleles of the apple gene that we previously talked about, the big B version and the big B version on that particular chromosome. We're ignoring all the other chromosomes, just looking at the B. Okay, so that's the allele. And it's diploid, so it's got two of the alleles, okay? Allele, and it's diploid. P2, let's designate as having two little b's, okay? It's also diploid, so it's got two of them, and that's the alleles that it has of the apple gene. Now let's think about meiosis and the products of meiosis in parent 1. In this case, it's pretty simple. You're big B, and so there's only one type of outcome, one type of gamete you're going to make after meiosis, and it is going to have the haploid genotype of B, big B. Okay? So that is the haploid genotype because gametes are always haploid. And for P2, the gamete is going to be little b, because that's the only option. There's only little b's, and it's only going to be one of them because, again, it's haploid. So we are now going to cross these parents to one another. When mating occurs, there is one male gamete and one female gamete that come together to make a diploid organism. They restore the diploid number of chromosomes. And so these two gametes will come together. And in the first generation offspring, there will be one big B and one little b. This offspring is diploid again. And 100% of those offspring will be big B, little b in their genotype. This is really important in genetics. And this is one way you can represent this type of thing, but there's actually a better way, and I'm going to show it to you on a slide. And the better way is called a Punnet square. And I'm going to make a note here, Punnet square representation. You'll have lots of practice of working with Punnet squares. But I needed to do this in longhand before you could look at a Punnet square. So let us look at this Punnet square. And actually, I want to make one other point before we look at the Punnet square representation. If you have two of the same alleles, big B, or little b, you get a special term, and that is called homozygous. If you have one big B and one little b, two different alleles, you are called heterozygous. So these big B, and little b are homozygous, and the big B, little b, of which 100% of the cross results in, those would be called heterozygous. And those are terms you should know. Now let's look at our slides. And here, in the Punnet square, you can see that there's a kind of formalization of how you represent things. The parents are represented along the side and the top of the square, and the gametes are represented next to the parent. So you kind of start with the parental genotype, and then you split it into the gametes that can form along the top and along the side. And then you do the cross. You say that gamete big B will match up with gamete little b and give you big B, little b. Gamete big B will match up with this little b to give you big B, little b, and so on. And you do this for every combination of gametes from each parent, filling in the squares with the outcome of the combination. Okay? It's logical and it's organized, and it's very helpful in figuring out the genotype and the proportions of the different genotypes. This is a simple example, and everyone, this is the same example that I did on the board, all of the offspring are big B, little b. But this is a different Punnet square representation of genetic crosses using genotype. Genetic crosses can also follow traits so that you can match up the genotype with the trait, or indeed just follow the trait and later on try to figure out the genotype. For example, let's have another example to extend our example. Let us have our parents and our genotype, and now we're going to add phenotype, another word for trait. Okay? And then we're going to have our first generation. Okay, so our parents again are P1 and P2, and they're going to cross. They're going to mate. Here is P1. We'll give it the same genotype as before, but the difference is we're now going to add a phenotype. Big B has a green phenotype. The apples are green. Little b has a yellow phenotype. The apples are yellow. When you cross those together, as before, you get out the F1. The F1 is big B, little b, and they are all green. Aha! So you started with green and yellow apple plants, apple trees, and when you crossed them, you came out with all green. In this example, green is said to be dominant, and this would refer to the B allele. Yellow is said to be recessive relative to big B. That would be the little b allele. So here are two terms, green and yellow, defining dominant and recessive. Which of the traits carries on into the next generation? Okay. What does this look like on a Punnet square? We can do the same example exactly, but now we can add phenotypes. And I'm going to do something a little tricky here, because in the example that I've given you, I've shown you actually an F1 cross. So let us keep that slide in our mind. But let's go back to the board just for a moment before you can really interpret the slide that I've put up. So we've followed the cross with various traits, but then what I've done to make it interesting and to show you some other principles is that I've done an F1 cross. In an F1 cross, you're taking the offspring from that initial parental cross and you are crossing them to one another. So here, the parents, the genotype and the phenotype, the parents would be the F1 crossed by the F1. And from our previous board, those F1 plants have the genotype big B, little b, both of them. And the phenotype they have is green. And now we can look and see how this cross reads out using a Punnet square, looking at a Punnet square. Okay, so let's now look at our slide where I've got this cross indicated. The parents are F1 with green phenotypes and the genotype is big B, little b. And you can see now that the gametes from each parent can be one of two kinds. They can be big B or they can be little b. Parent 1 can give big B and little b, parent 2, big B, little b, okay? And when you fill in your Punnet square, you can see that there are actually 3 different genotypes. There is big B, big B, little b, and little b, okay? 3 genotypes. There are only 2 phenotypes. There is green or there is yellow, okay? 2 points from this. Firstly, you can see that genotype does not always go with phenotype. The big B and the big B, little b have the same phenotype, but they've got different genotype. And the little b has a different phenotype to the genotypes that have got a big B. And this again is an example of how we know that the big B allele is dominant to the little b allele. You only get yellow apples when you've got 2 little b's. When you've got any one big B, you're a green apple. Okay? So this is a Punnet square representation that has both genotype and phenotype. And then the last thing I want to tell you, which I'll put on the same board here, is something called a test cross. You may not know what the genotype of the parents are. You might want to figure this out. You might think, are the parents big B, or big B, little b? You can figure this out if you cross those parents to something that is a true breeding strain. And a true breeding strain in this case will be a homozygous recessive strain. So you're going to cross this to little b, which is homozygous. It's got 2 of the same alleles, and it is recessive, and it is called true breeding, where true breeding means that if you cross little b to itself, you'll come up with yellow all the time. You'll never suddenly start getting green. True breeding means you know what you have in your breeding stock. So this notion of a test cross is something you can use to figure out genotype. And here's a slide that has got a test cross where you're not sure if it's big B or big B, little b. You make 2 separate, or you make a number of crosses. And I've shown you 2 outcomes. If parent 1 was big B, all of your offspring would be green. If parent 1 was big B, little b, half of your offspring would be green and half of them would be yellow. And you can follow that on the Punnet square with the genotypes associated. Okay, so we've talked now a bunch about monohybrid cross, and you can go and look at the class exercise and practice some Punnet squares on the class exercise to cement these kinds of concepts.